Hello everybody. I hope I am audible to all of you. Just give me a nod whether I am audible to you or not. Okay. Then we will start the class directly. Hello everybody. Audible? Just give me a nod. Okay. So, good evening to all of you for joining the session. In this session, we are going to discuss only a small topic, that is the spatial sense in smell and taste. These two spatial sense we are going to discuss. Okay, right. So, before starting the session, you know that this spatial sense, that is the smell and taste, this is an extra topic which is not included in my app, that is the Physiology Mind, right? So that's why this topic will be beneficial for those students who are using my app, and it will also be beneficial for others who are also not using my app. So this topic is one of the important in Central Institute examination like AIIMS and GIMR. Also this is important for the NEET examination, right? So spatial sense, whenever we are talking about the spatial sense, we know why this is known as the spatial sense, right? So first of all, we are starting from here, that what is the meaning of the spatial sense, why it is spatial, what is so special in these senses. So always remember that spatial sense means for this kind of sensation there is a specialized organ devoted to this kind of sensation. Like for example, if you think of pain, temperature or touch, there are receptors which are not specialized for only touch, or there are receptors which are not specialized for only pain, and those receptors are present throughout your body. But if you think of these spatial senses like vision or hearing, then you will find out that these rods and cones, they are only specialized receptors which are present at the level of retina. Similarly here, for the hair cells, which are the receptor for the hearing and balance, they are also located in a specialized area, and only they are responsible for some special function. Similar for taste and smell sensation. That's why we put all of this sensation under the category of spatial, means they are specially devoted for some kind of sensation, not for generalized sensation. They are not present in general part of the body. Now in today's class we are going to discuss these two, taste and the smell, that is the gustatory and the olfactory sensation. Now before going into this two main topic, all of you know that the vision, that is the rods and cone, they are under the heading of the photoreceptors. So they are nothing but photoreceptor cell. All of you know this thing. Then if I ask you what about this hearing and the hair cells which are maintaining your balance, you know these are the mechanoreceptor. So they are nothing but your mechanoreceptor. Why mechanoreceptor? Because you know that whenever sound is causing vibration of the basilar membrane, there will be movement of the hair, and because of the movement of the hair, what will happen, there are some mechanosensitive or stress sensitive channel that will open up, and that will leads to sound transduction. Very simple. But on the other hand, if you look at these two spatial senses that we are going to discuss today, they are nothing but chemoreceptors. So this is nothing but the chemoreceptor. So smell and taste, both of them are chemoreceptor. So whenever we will see the detail about the mechanism of the receptor, you will find out that these receptors are stimulated by chemical signals. That's why they are under the heading of the chemoreceptor. But don't confuse with this chemoreceptor with the chemoreceptor at the level of carotid body and aortic body, because they are for different purpose, and this chemoreceptor for sensation, this is a kind of sensory chemoreceptor, right? So first we are starting from the olfaction, that is the smell, sense of smell or olfaction. Now look at, all of you know that sense of smell is perceived by a area which is located at the roof of our nose, right? So this roof of our nose contain a area which is known as the olfactory epithelium. So note down here, the olfactory epithelium has a area of 3 into 3 centimeter square. Okay, Genong is saying that the area of this olfactory sensory epithelium is around 10 centimeter square. So I am saying 3 into 3, that means 9 centimeter square is the area. Now what is there inside this olfactory sensory epithelium? If you find out, then look at here. So we have magnified this area. If you magnified this area, what you are finding out? So first of all, you note this cribriform plate of the ethmoid bone. Okay, so just below this cribriform plate, what you are finding out, this is nothing but your olfactory epithelium, and inside this olfactory epithelium you are finding out that multiple types of cells are there. What are the multiple types of cell? Number one, olfactory sensory neuron. This is an MCQ. So first, this is an MCQ, olfactory sensory neuron. So number one cell is the olfactory sensory neuron, and if you look at this olfactory sensory neuron, you are finding out that they are a kind of bipolar cell. So first MCQ is nothing but olfactory sensory neuron are a kind of bipolar sensory neuron. Means the receptor for the olfaction is located on a neuron which are a type of bipolar neuron. Then apart from this bipolar neuron, you know, there are some supporting cells also, and there are some basal stem cell also. So I have to remember three things, that olfactory epithelium is consist of three different types of cell. One is the olfactory sensory neuron, second is the supporting cell, and third is the basal stem cell. So from the name basal stem cell means you know that this is the cell which can regenerate the bipolar sensory neuron when it is dead. That means this bipolar sensory neuron, they have a lifespan of, you know, that they can survive only for 1 or 2 months. So this is an important information. Bipolar sensory neuron for olfaction, they survive only for 1 to 2 months, right? So after the death of this bipolar sensory neuron, this basal stem cell can mitotically divide and give rise to the bipolar sensory neuron. That's why they are known as a basal stem cell, right? And third is the supporting third one is the supporting cell. Supporting cell is responsible for secretion of mucus. Okay, why that is required? Look at, if you look at this bipolar cell, then you will find out one important thing, that the dendrites of this bipolar cell are covered by mucus layer, and this mucus is secreted by supporting cell. So why this covering is important? You know that Genong is saying that this bipolar neuron of olfactory epithelium, they are exposed to the external environment. This is the only place where the neurons are directly exposed to the external environment. You know that everywhere in our body neurons are covered up by protective covering, but this is one area where the bipolar neuron are exposed to the this nasal epithelium, okay, this olfactory epithelium. Because of the exposure, what will happen, there is a covering by this mucous membrane, and that is secreted by supporting cell. So three things: bipolar cell, that is the olfactory sensory neuron; stem cell, that give rise to bipolar cell; and there is supporting cell. Supporting cell is responsible for secretion of mucus. Then also note down that the axons of this bipolar cell, they are piercing through this cribriform plate, and they are merging at a area. This area is known as the olfactory bulb. This is known as the olfactory bulb, right? So bipolar sensory neuron extended up to the level of the olfactory bulb. Now if I magnify this one of this bipolar sensory neuron, what I am going to see? So look at here. So here I have given a magnified view of a single bipolar olfactory sensory neuron. Getting my point? So this is also known as the first order neuron. So this is the dendrites, and this is the axon part of this bipolar neuron. At the top of these dendrites, you are finding out that there are presence of cilia. How many cilia are there? So at the top of these dendrites, there are 6 to 12 cilia are present, and at the level of this cilia, you are finding out this red colored dot. What are these red colored dots? These red colored dots are nothing but they are your odorant receptor. They are nothing but your odorant receptor. That means you are understanding here that the dendrites contain cilia, and the top of this cilia there are presence of odorant receptor, and this receptor, whenever it is stimulated by some chemical signal, suppose this is the chemical signal, whenever it is stimulating this receptor, the receptor potential will be generated at the level of this bipolar neuron, and that receptor potential will travel through this axon, and axon will produce action potential, and that action potential ultimately leads at the level of olfactory bulb. So what I am saying here, this is nothing but olfactory bulb. What is there inside the olfactory bulb? At the level of olfactory bulb, this first order neuron of olfactory pathway is going to make a synapse with the second order neuron of the olfactory pathway. What is the name of this second order neuron? The name of this second order neuron is the mitral cell. This is also known as the tufted cell. So there are two types of second order neuron. It may be mitral cell, which is the largest cell, and the smaller size cell are known as the tufted cell. So this mitral and tufted cell, they are nothing but the second order neuron of this olfactory pathway, and this second order neuron will directly end at the level of the cortex. So we'll come to this pathway a little later. So you can find out 2-3 peculiarities here. What the peculiarities? You are getting that only 2 neurons are required for the olfactory sensation to reach up to the level of cortex. But compare with your pain sensation, temperature sensation, everything require at least 3 neurons from the periphery to the level of cerebral cortex. But here you are requiring only 2 neurons. First order neuron here and the second order neuron here, that is the mitral or tufted cell. Second thing is that the second order neuron is not crossing to the opposite side of the body. That means it's an uncrossed tract. So olfactory tract is an uncrossed tract, right? And third is that whenever it is reaching from this olfactory bulb to the level of olfactory cortex, it is bypassing the thalamus. Means we are not getting any relay station at the level of thalamus before reaching up to the level of cortex. Although you know that after going to the cortex, it will go to the other part, and from there it may go to the level of thalamus, and from thalamus again it will reach to a different part of the cortex. We will discuss this thing a little later, okay? But please remember that when we are using that olfactory pathway bypasses the thalamus, it means that second order neuron is directly reaching at the level of cortex without relay at the level of thalamus. Although it may go to the level of thalamus later on, but this is not the second order pathway. It is not going there thalamus. It is going to the cortex before reaching the thalamus, right? So what is the bottom line from here? Bottom line from here is the bipolar neuron contain the odorant receptor, okay? And odorant receptor, if it is stimulated, it will produce receptor potential here, and that receptor potential will generate action potential here. Action potential will travel to the olfactory bulb. At the level of olfactory bulb, the first order neuron is making a synapse with the second order neuron. What are the second order neuron? It may be mitral cell or it may be tufted cell. And the junction between this first order and second order neuron, this synapse, this is known as glomeruli. This is known as glomeruli. Because of the structural similarity of this synapse with that of the glomerulus, this is known as the olfactory glomeruli. So from this olfactory glomeruli, the second order neuron is directly reaching at the level of cortex. Now, one of the most important thing that is asked in Central Institute examination is that how this odorant receptor is stimulated by chemical signal when it is reaching at the level of your nose. So what is happening at the level of this receptor, that we are going to discuss now, right? Okay. So to understand what is happening at the level of receptor, I am drawing one cilia. So this is nothing but a cilium, right? Because I told you that on the top of the dendrites there are 6 to 12 cilia. So I am drawing only one of this cilia, and this cilia contain, so this is nothing but the odorant receptor. This is nothing but receptor. Fine. Now always remember, this is the first MCQ again. It's a G protein coupled receptor. It's a G protein coupled receptor. So odorant receptor are G protein coupled receptor. What will happen? Whenever some chemical molecule or odorant molecule is attached with this receptor, it will stimulate the G protein. And whenever G protein is stimulated, you know the downstream pathway, that it is the G protein will be activated. Activation means the alpha subunit will be separated out, and that alpha subunit will produce a second messenger, that is nothing but CAMP. Now note down the next part. This CAMP, now it is going to open up one particular ion channel, which is very, very important. This ion channel is a non-specific cation channel which are present on the cilia, through which calcium will enter, through which sodium will enter, through which small amount of potassium can also enter. Okay, so what I am saying, calcium will enter, sodium will enter, and through this channel the potassium may also, the channel is also permeable to potassium also, right? Okay, potassium will not enter, sorry, potassium will go out, but that amount of going out of potassium is very, very less. But what is the main ion here? The main ion is the entry of this calcium. So that's why I am saying that this channel is a non-specific cation channel. Non-specific means calcium is also entering, sodium is also entering. That's why this is known as the non-specific cation channel. Because of the entry of this calcium and sodium, what will happen to the cilium? There will be depolarization of the cilium. So I am writing here that there will be depolarization. Now whenever I am saying that there will be depolarization, then obviously the query will be that what is the RMP of this? What is the RMP of this olfactory sensory neuron? That is the bipolar neuron. What is the RMP? That is minus 55 millivolt. Now if this depolarization is sufficient to raised up to the level of minus 35 millivolt, then this depolarization is sufficient to produce one action potential. And this depolarization, which is occurring at the level of this receptor neuron or sensory neuron, this is known as the receptor potential generation. Receptor potential generation. Note down, please note down. So what is the main ion for the receptor potential generation? Calcium ion, although sodium is also there. Because of these two positive ions, there is depolarization, and this depolarization is known as the receptor potential generation. But this is not all. Note down the most important thing. This calcium can further stimulate another ion channel that is known as calcium stimulated chloride channel. It is known as the calcium stimulated, this is known as the calcium stimulated chloride channel. Very, very peculiar, very, very peculiar. So whenever this calcium stimulated chloride channel is open, note down the most important thing, most peculiar thing. This has been added up in recent edition of Genome. The chloride will go out of this cilia. The chloride ion is going out of the cilia. This is peculiar because in other type of cell in our body, we know that chloride concentration is high outside. That means whenever it is open, chloride channel is open, the chloride enter inside the cell. But olfactory bipolar cell, they contain huge concentration of chloride, and that concentration is high inside the cilium or inside the cell. Because the chloride concentration is very high inside the cell, what will happen? This chloride is going out of the cell, and whenever the chloride is going out, I can understand like this. Chloride means negative charge. So negative charge is going out of the cell means cell is losing negative charge. So if the cell is losing negative charge, what will happen? It will be further positivity. That means I can say it will leads to further depolarization. It will leads to further depolarization. What do you mean by that? You are saying, sir, you are saying that depolarization has already occurred, and if this depolarization is sufficient enough, then it can produce action potential. Then what is the need of this second chloride channel opening? This is known as the amplification of this signal. This chloride channel opening is responsible for amplification of the signal. What do you mean by that? If there is only calcium channel open or sodium channel open, then the action potential will occur, but the voltage of this depolarization voltage may not be very high. But whenever there is additional chloride channel open, because the chloride is going out, further depolarization will occur, and that will amplify the signal. Because of this amplification of the signal, what will happen? The action potential frequency will be increased, and whenever there is increase in action potential frequency, what will happen? Even if the amount of signal or amount of smell, amount of chemical smell is very, very small, it can be easily detected by the neuron, because neuron is amplifying the signal. So that's why it is very, very important to remember this special channel, that is calcium stimulated chloride channel. Not only this bipolar sensory neuron for olfaction, remember this calcium stimulated chloride channel is also present in photoreceptor, that is at the level of retina. It is also present in some of the smooth muscle, and it is also present in some of the epithelial surface. So 4 areas I have to remember this special type of chloride channel. Okay, calcium stimulated chloride channel. One is the bipolar sensory neuron of olfaction, second is the photoreceptor cell, third is the smooth muscle, and fourth is the sum of the epithelium cell. And everywhere this channel is responsible for amplification of the signal, which will be helpful for the action potential generation. Now you got it. So now we came to know that at the top of this cilia, okay, at the top of this cilia where the odor and receptor are present, when this odor and receptor are stimulated, what is going to happen? There will be receptor potential generation. If I ask you what is the main ion for this receptor potential generation? Calcium, although sodium ion is also there. Then apart from this calcium and sodium ion, there is another ion, which is the chloride ion, which is responsible for amplification of this signal. And after this signal or receptor potential is generated, then the action potential will be generated, and the action potential will reach to the level of this olfactory glomeruli or olfactory bulb, and this second order neuron will be stimulated, and the second order neuron will bring the sensation to the level of cortex, and the perception of olfaction will be detected by the level of cortex. Now what are the different parts of the cortex? I am coming there a little later, but before that, one of the important information I have to remember, that is the information which has been asked in AIIMS entrance examination system. AIIMS entrance examination, they have asked this question. What is that? We have just discussed that olfactory receptor, that is the odor and receptor, are present at the level of cilia. There are 1000 olfactory genes are there in human, which are coding this olfactory receptor. Getting my point? What I am saying, that this olfactory receptor that we have discussed, they are coded by different genes. How many genes are dedicated for this olfactory receptor coding? Thousands genes are there, and this is the largest family of human genomes known so far. This was the question which has been asked in AIIMS entrance examination, that which of the following is the largest family of human genome, that is the olfactory receptor coding genes. How many genes are there? 1000 genes are there. But all of these 1000 genes are functional? Answer is no. All of these 1000 genes are not functional. Only 4 of these 1000 genes are functional, means rest of the genes are pseudogenes, means only these 400 genes, they code the protein which are responsible for generation of the formation of this odor and receptor. So only 400 genes are functional. Some of the books are saying 350, and some of the books are saying 400, but I have taken this from Genau, so remember that. So within this 1000 olfactory genes, only 400 genes are functional. Discovery of this olfactory receptor gene family was done by these 2 scientists, that is the Linda Buck and Richard Axel, and because of this discovery, they were awarded the Nobel Prize in Physiology and Medicine in 2003, right? So this is one of the important information you must have to remember. So after this, now we are entering at the level of olfactory bulb connection. So I'll come to this diagram. Before that, I will try to simplify this olfactory bulb connections. What is the simplification? Again, quickly revision. So this is bipolar cell, right? Bipolar cell axons is entering at the level of olfactory bulb. So I am drawing another bipolar cell. So this is another bipolar cell, right? So again the axon is entering at the level of olfactory bulb. Okay, so at the level of olfactory bulb, it is making a synapse with the second order neuron. If the neuron is large enough, then it will be known as the mitral cell. If the neuron is small enough, then it will be known as the tufted cell. So these are the second order neuron of the olfactory pathway, right? Now this area where this first order and second order neuron are making synapse, this is nothing but olfactory glomeruli. So this is one glomeruli, this is the second glomeruli, and all of these glomeruli, around thousands such glomeruli are present inside a single olfactory bulb. So this is your olfactory bulb. Inside this bulb, around 1000 such glomeruli are located. Now, at the level of this glomeruli, apart from this first order and second order neuron, there are two other neurons. One is the granule cell and one is the periglomerular cell. So one is the periglomerular cell. What is mean by that? Periglomerular cell, they will be present here. They will be present here. So this is the cell body of the neuron, periglomerular cell, and this is the axon and dendrite part of the neuron. So this is known as the periglomerular cell. What type of cell this is? Periglomerular cell, and the other cell is the granular cell. So suppose this is the granular cell I am drawing, I am writing G, granular cell. Both of them, so periglomerular cell and the granular cell, both of them are inhibitory cell at the level of olfactory bulb. So whenever I am saying inhibitory cell, that means the neurotransmitter produced by them is a GABA. Now how these cells are functioning? So to understand that, I am just wiping out this glomeruli boundary. Now look at, so these are the dendrites of the second order neuron and these are the dendrites of the second order neuron. What this granular cell is doing, the granular cell, it will be connected with the dendrites of one second order neuron with the dendrites of another second order neuron, and this connection is by only dendrodendritic connection. What I mean? Don't understand. Means this granular cell is a special type of cell. You must have to remember. Why it is special? Because this granular cell neuron, it does not have any axons. So granular cell means it contain only and only dendrites. That means the dendrites, now you look at the diagram, the dendrites of this granular cell is making a junction or synapse with the dendrites of the mitral cell. Again here, the dendrites of the granular cell is making a synapse with the dendrites of the tufted cell. So what type of synapse this is? This is a kind of dendrodendritic type of synapse. Getting my point? In the central nervous system, you must have heard that there is axodendritic, axosomatic, axoaxonic type of synapse. But this dendrodendritic synapse is very, very rare, and it is present only at the level of olfactory bulb. The junction or synapse between this granular cell with that of the second order neuron, that is the mitral and tufted cell, this synapse is a dendrite synapse. That's why this is known as the dendrodendritic synapse. So how this granular cell is functioning? Suppose you have stimulated this bipolar sensory neuron here. Stimulation is there. So stimulation means action potential is traveling through. So it is going to stimulate the second order neuron of the mitral cell, second order neuron of the olfactory pathway, that is the mitral, or it may be stimulating the tufted cell. Right. So suppose only one of these two pathway is stimulated. Only one of these two pathway is stimulated here. Okay. So this neuron is stimulated. So whenever this second order neuron or this blue color neuron is stimulated, now it is going to stimulate this granular cell here. Now whenever the granular cell is stimulated by this mitral cell, now this granular cell will release the GABA neurotransmitter here, and by releasing the GABA here, what it is going to do? It is going to inhibit the tufted cell. This tufted cell is the nearby tufted cell or the surrounding tufted cell. This kind of mechanism is known as the lateral inhibition. This is known as the lateral inhibition by this granular cell on the second pathway. What is the importance of this lateral inhibition? So where is your stimulus? Your stimulus is going through this neuron only. The brain has to perceive that what is the particular type of stimulus depending on the identification of this type of neuron. So this neuron is stimulated, but the other neuron is not stimulated. But if the surrounding neuron is not inhibited, then this signal will not be serpent. The signal will not be prominent. So that's why by inhibiting the surrounding pathway, you are focusing only one type of signal. The brain is focusing only one type of signal by inhibiting the nearby neuron. Okay, this is known as the sharpening of signal. This is nothing but contrast enhancements. So this lateral inhibition by this granular cell, a similar mechanism is also performed by this perigranular cell. This is responsible for contrast enhancements of olfactory signal. Now everything is written at this picture which I have taken from a book. You can find out now all of this discussion. So this is the olfactory sensory neuron. This is the first order neuron coming. This is the first order neuron coming, and it is making a second order neuron connection. This is the mitral cell, and suppose this is the tufted cell. So at the level of this glomeruli, I can say this is the periglomerular cell, and at the level of this dendrodendritic junction, you can say that this is the granule cell. So this granule cell and this periglomerular cell, what are they? They are the inhibitory neuron. Inhibitory neuron means what kind of neurotransmitter they are going to produce? They are going to produce the GABA neurotransmitter. But between these two inhibitory neuron, I must have to remember that the granule cell is special because the granule cells have no axons. It makes synapse with the second order neuron only by this dendrodendritic pathway. And because of this type of inhibition, what will happen? The neuronal signal will be sharpened. Or you can focus on the olfactory signal. Getting my point? And then we know that the dendrites, the axons of this mitral cell, or the axons of this tufted cell or mitral cell, whatever, they are forming the second order neuron of the olfactory pathway, and they will directly enter at the level of cortex bypassing the thalamus. So now look at the cortical connection. And again, this is one of the important thing to remember. So this is the olfactory bulb. So what we have discussed? We have discussed that olfactory bulb contain multiple olfactory glomeruli. So you can find out this is one glomeruli, this is another glomeruli, and from this olfactory glomeruli, the second order neuron, axons of this second order neuron is forming the lateral olfactory tract, and this tract is reaching directly at the level of cortex. At least you have to remember 5 different cortical area for olfaction. Okay, now these are the 5 different cortical area. What are the different area? You can see from this diagram that the cortical area, various cortical area for olfaction is the anterior olfactory nucleus, right? Okay, olfactory tubercle, piriform cortex, amygdala, and enterorenal cortex. Note down very carefully, very important. So I am coming to this function of this area, but before the function of this cortical area, you note down another things, that some of the olfactory pathway, like for example from this olfactory tubercle, from this piriform cortex, the fiber is also entering at the level of thalamus, and from the thalamus again it is going to a different area of the cerebral cortex, that is nothing but orbitofrontal cortex. This pathway, which is going from the level of thalamus up to the level of this orbitofrontal cortex, according to Guyton, this is known as the new pathway for olfaction. This is known as the new pathway for olfaction. So what is the new pathway for olfaction? The pathway which is reaching at the level of this thalamus. Generally, the second order neuron is reaching to the level of the cortex without relay at the level of thalamus. Okay, so that's why we say that olfactory pathway is an exception. It is an uncrossed fiber, and it takes 2 fiber only, 2 neuron only, 1 neuron from the epithelium to the level of bulb, and from the bulb to the level of cortex, 2 sensory neuron only. And third is that it is not relaying at the level of thalamus, so it is directly reaching up to the level of this cortical area. But some of the fiber from this cortical area, it is reaching at the level of thalamus. Which nucleus of thalamus? Important for MCQ. AIIMS PG has already asked. This is nothing but dorsomedial nucleus of thalamus. medial nucleus of thalamus, and from the dorsomedial nucleus of thalamus it is reaching at the level of orbitofrontal cortex. Orbitofrontal cortex, this pathway which is reaching from this thalamus to the level of orbitofrontal cortex, this is known as the new pathway for olfaction. This new pathway for olfaction is particularly dedicated for olfactory discrimination, means this orbitofrontal cortex is responsible for discrimination between two different types of odor. We can discriminate millions of odor, but this is the function of this orbitofrontal cortex, because of which we can discriminate two different types of odor, right? So what is the important thing? I have to remember the function of all of this cortical area along with this orbitofrontal area. That is the most important for your central institute examination. So what are the different cortical area? The piriform cortex, it is responsible for perception of smell. You can perceive the smell that something is there, but you cannot differentiate to smell until and unless you take help of this orbitofrontal cortex. So perception of smell is by the piriform cortex, but the conscious discrimination of the odor is done by the orbitofrontal cortex. Along with that, the amygdala is also responsible for olfaction. What type of olfactory perception? That is for the emotional response of olfactory stimuli. You know that odor, smell, it is attached with the emotions, and that's why the perfume company are making big business because of this emotional attachment with the olfaction, right? And which part of the brain area is responsible for this emotional component? That is the amygdala. And apart from this, entorhinal cortex is also responsible for some part of the olfactory, and that is related to olfactory memories. You can remember that this is the smell which is typically you find out from these substances. So this kind of olfactory memory is because of the entorhinal cortex, right? And I already told this point that the fiber which is passing through the mediodorsal or dorsomedial nucleus of thalamus to the level of orbitofrontal cortex, this is known as the new or newer pathway of olfaction. Got it? So this is the basic thing. This is the basic thing that you have to remember about olfactory pathway. Uncrossed pathway, taking only two neurons, first order neuron and second order neuron, and bypassing the thalamus. For MCQ purpose, this area, functional areas of this cortex are very, very important. Now, if I look back at this diagram, I can find out that something is written here. That is nothing but vomeronasal organ. It is also attached to this olfactory bulb, but this is a special area of the olfactory bulb which is known as the accessory olfactory bulb, vomeronasal organ. Do you think this vomeronasal organ is present in human also? No. You must have read in biology that the vomeronasal organ, this is for detection of the pheromones. This is present in some of the lower mammals, but not in human. The neurophysiology books is saying that vomeronasal organ is rudimentary in human. It is not present in human, right? But although vomeronasal organ is not present in human, although the pheromones is not present in human, but Genome is writing two important things. That sense of smell or the olfactory sense is more acute, more strong in case of female. Okay, they can perceive more smell, more acute smell are perceived by female, women rather than male. That's why the perfume company, whenever they give some advertisement, always you will find out that the main is using the perfume and the female is attracted to that perfume. Why this kind of advertisement? Because the females are more, okay, they can perceive the sense of smell more acutely than male, and the perception of smell in case of female subject is more stronger at the time of ovulation. At the time of ovulation, the perception of smell is more stronger in female at the time of ovulation, right? So these two things you have to remember. So you can correlate many things, that if the perception of smell is very strong in and around the ovulation period, then what is going to happen, and that is the basis of this the X effect. Okay, so this is all about the brief discussion of your perception of smell or olfaction. Now we are coming to, if you have any queries, I'll come to that at the end of the topic, but before that we are going to discuss the second one, that is the sense of taste, right, or gustatory sensation. Now sense of taste, all of you know that the specialized organ is located at the surface of this tongue, right? So all of you also know that at the surface of this tongue, what are there? There are some little elevated area which are known as the papilla, What are the different types of papilla present on the surface of this tongue? They are fungiform papilla, they are foliate papilla, and they are circumvallate papilla, right? Now this fungiform, foliate and circumvallate papilla, they are located at a particular area of our surface. So this anterior part of this tongue, they contain mainly this fungiform type of papilla, right? At the foliate papilla, they are located at the lateral edge of this tongue, and at the base of this tongue, this V-shaped area, they contain mainly this circumvallate type of papilla. If you look at the papilla, what you are going to find out? You are going to find out that there are multiple taste buds. So this is the papilla. At the top of this papilla, you can find out there are taste buds. How many taste buds are there? In a single fungiform papilla, note down, in a single fungiform papilla, up to 5 taste buds are located. In a single foliate papilla, up to 100 taste buds are located, and in a single circumvallate papilla, there are around 100 taste buds located. If you have to choose only one answer, that which of these papilla contain maximum number of taste buds, then this is the best answer. And if you add up all of this number of papilla, then in human adult, there are altogether total 3,000 to 10,000 this kind of taste buds are there. Okay, there are 3,000 to 10,000 papilla are there. There are 3,000 to 10,000 papilla are there on the surface of this tongue, right? So 3,000 to 10,000 taste buds are located on the surface of this papilla, if you add up all of this. And also remember, in case of children, this number is more than that of the adult, right? So if you count all of these taste buds, how many taste buds are there? There are around 3,000 to 10,000 taste buds are there, and these numbers of taste buds are more in case of children. Now, is there any particularity that particular type of taste bud is specialized for a particular type of taste? Answer is no. Previous concept was that a different type of taste like sweet, salt is detected by different region of the tongue. But nowadays they are saying, Genong is writing, that any kind of this taste bud, any taste bud can detect any kind of taste. Means if there is a taste molecule, if there is a chemical signal, taste-tannin molecule is present on your surface of the tongue, what will happen? It can activate any of this taste bud. It can activate any of this taste bud. It may be sweet, it may be sour, it may be anything. It can activate any of this taste bud, right? So that you have to remember, that there is no particular location that the sweet will be detected here, salt will be detected, and no. Now the second thing that you have to note down here, all of you know this thing, I hope, that the anterior 2/3 of the tongue is supplied by this chorda tympani nerve of the facial, chorda tympani branch of the facial nerve, and the posterior 1/3 is supplied by the glossopharyngeal nerve. So these are the two nerves which are responsible for detection of your taste sensation from the tongue. Anterior 2/3 is the chorda tympani, posterior 1/3 is the glossopharyngeal. Apart from that, you also know that some of the taste buds are also located at the level of pharynx, and the nerve fiber which is dedicated for the pharyngeal taste sensation is nothing but the vagus nerve or the 10th cranial nerve, right? But apart from all of this, one important information for MCQ, that you know that trigeminal nerve, what is the 5th cranial nerve, it also has a sensory component which also supply the surface of the tongue. And whenever you take some spices food, like if you take some chili, You know that there is an ingredient inside the chili which is nothing but capsaicin, right? Or if you take some kind of hot water or hot tea, what will happen? You can easily perceive this sensation of capsaicin, that is the chili sensation of chili, or you can perceive this hot sensation. But this is not a kind of taste sensation. This is not going to stimulate your taste bud. It is going to stimulate your trigeminal nerve, because the ending of the trigeminal nerve, which is present on the surface of this tongue, it contains a special ion channel which is known as the TRPV1 ion channel, that is the transient receptor potential channel. This ion channel we have discussed in pain pathway, in pain receptor pathway, that this is the receptor channel which is also responsible for pain sensation. So if you give this capsaicin, you can also perceive pain. If there is presence of capsaicin on the surface of your tongue, you can find out that it is producing a burning sensation of the tongue. Or if there is hot sensation, you can also perceive the hot sensation at the surface of this tongue. By which receptor? By this TRPV1 ion channel. And this TRPV1 ion channel is present on which neuronal ending? This is present on fifth cranial nerve ending, that is nothing but the trigeminal nerve. That is important to remember, because these are not the taste sensation. Whenever you are getting some burning sensation or hot sensation inside the mouth, that is because of this TRPV1 ion channel activation. Now we are entering inside the taste bud. So there are around 3000 to 10,000 taste bud. Now we are focusing only one taste bud among all of them. So now we are focusing on one taste bud, what we are going to get. So this is the diagram of a single taste bud. What are we finding out? Now inside the taste bud, there are around 100, there are around 50 to 100 taste cells are there. So this is the first thing that you have to note down, that inside a single taste bud, there are 100, around 50 to 100 taste cells are there. Different types of taste cells are there, type 1, type 2, different, but you don't have to remember detail about this. You just remember there are taste cell, along with that there are basal cells. So the second type of cell that you have to remember, that is the basal cell. We have also seen this basal cell at the level of olfactory epithelium. Recall, the basal cell at the level of olfactory epithelium, they behave like a stem cell. That means they can regenerate the olfactory sensory neuron. Same thing here. This cell can regenerate the taste cell. So this can regenerate the taste cell. That means this basal cell is also acting like a stem cell. Although we do not use the word stem cell, but it can regenerate the taste cell. Why? The half-life or the lifespan of this taste cell is only around 10 days, only around 10 to 12 days. Because of this because of the lifespan of 10 to 12 days, what will happen? After 10 to 12 days, when the cell is dead, then the basal cell will regenerate the new taste cell, right? And also note down that at the top of this taste cell, there are some microvilli, and these microvilli are exposed to an area on the surface of your tongue. So this is the surface of your tongue, which is covered up by the saliva, and this is known as the taste pore. Means whenever certain kind of chemical like sugar or sour taste or any kind of chemical signal is present here, it is going to bind with this microvilli portion of the taste cell, and that will leads to depolarization of the taste cell. Depolarization of the taste cell is known as the receptor potential generation at the level of taste cell. Whenever it is generating receptor potential, it is going to produce a neurotransmitter, that is glutamate, and that neurotransmitter is ultimately going to activate this neuron, and this neuron will produce action potential, and this action potential will ultimately reach to the cerebral cortex through the nucleus tractus solitarius and through the thalamus. So what I am saying? I am saying here that this pathway of taste sensation or gustatory sensation, they are not bypassing thalamus like that of the olfactory pathway. It is going through the thalamus. First, this neuron, first it will reach to the nucleus tractus solitarius of the medulla, then it will reach at the level of thalamus, then from thalamus it will reach at the level of cortex. Again, it is taking help of 3 neurons, not like that of the 2 neuron of olfactory pathway. And on the right hand side, what you are finding out? On the right hand side, you are finding out the electron microscopic picture. You can easily find out this is asterisk marks. They are nothing but the cross section of the different types of taste cell, and this arrowhead, they are nothing but the sensory neuron. They are nothing but the sensory neuron which you are finding out here. So bottom line, on the surface of your tongue there are papilla. Inside the papilla there are multiple taste buds. Inside the taste bud there are taste cells, and this taste cell contain microvilli. At the microvilli level there are presence of receptor. So now if I magnify one of these taste cell from this bud, then you will find out that the cell look like this. So there are 2 variety of taste cell. The detail is not required for your purpose, but you have to remember that salt and sour, they are detected by a different types of taste receptor, and the sweet, umami and bitter, sweet, umami and bitter, they are detected by a different type of taste receptor. Getting my point? The bottom line is that salt and sour, the receptor which are responsible for detection of salt and sour, they are ion channel receptor. Means the receptor itself is an ion channel. Suppose this is an ion channel and it is behaving like a receptor, means it will attach with the chemical substances which is producing the taste sensation, and when the ion channel is stimulated by this chemical signal, it will open up and some ion will enter. After the entry of this ion, ultimately calcium is going to enter, and this calcium is going to produce the receptor potential, and this receptor potential is ultimately going to generate the action potential, and the action potential will travel through the sensory afferent neuron, which may be 7th cranial nerve, that is the facial nerve, chorda tympani branch of facial nerve, or it can be of the 9th cranial nerve, that is the glossopharyngeal nerve. On the other hand, sweet, umami and bitter, the receptor which is dedicated for these 3 kind of taste, this receptor are G protein coupled receptor. G protein coupled receptor, like that of the olfactory receptor. G protein coupled receptor, like that of the olfactory receptor, right? So this G protein coupled receptor means whenever the chemical is attached with this receptor, it will activate the G protein, and through this G protein it is going to stimulate various mechanism inside the cell, which will ultimately lead to influx of calcium, and this calcium is ultimately going to produce the action potential on this cell. So I don't have to remember detail about all of this, but I must have to remember that salt and sour are detected by ion channel type of receptor. This kind of receptor is also known as ionotropic receptor. And sweet, umami and bitter, they are detected by G protein coupled receptor. G protein coupled receptor are also known as metabotropic receptor. Getting my point? Ionotropic receptor and metabotropic receptor. Metabotropic receptor means if the receptor is coupled with the G protein, wherever your G protein coupled receptor are there, they can be also renamed as metabotropic receptor. And if the ion channel is behaving like a receptor, they are the ionotropic type of receptor. So most important thing, salt and sour, ionotropic receptor. Sweet, umami and bitter, they are metabotropic receptor. Now for Central Institute examination, you must have to remember detail about this receptor, because if they are asking tough question, they are going to ask you question from this receptor detail. So this is salt and this is sour. Which kind of receptor? They are ion channel receptor. They are ionotropic receptor. Then sweet, bitter, umami, they are metabotropic receptor or G protein coupled receptor. You can find out this diagram. Here there is no ion channel like diagram, but here this is ion channel through which ion can enter. That's why it's a kind of ion channel receptor. This diagram is taken from Genome, but here the receptor will be attached with the G protein. It will be attached with the G protein, right? So 5 basic taste sensation, we all know this thing. Salt is nothing but the taste of NaCl, all of you know. Sour Sour is nothing but it is the taste of H plus ion. If you give one drop of diluted HCl on the surface of your tongue, then the sour taste is going to be produced. That is the basic sensation. Sweet, you know, this is the taste of sugar, okay, that is a sucrose, okay. Then bitter is nothing but the taste of like quinine. If you give a drop of quinine, that is the bitter taste. And umami, important. What is umami? Umami, it's the fifth primary taste sensation. It is the taste of a salt which is known as this umami receptor is stimulated by a chemical which is nothing but monosodium glutamate. So it is a salt. Monosodium glutamate is a salt. This salt is commercially available as the name of Ajinomoto. You must have had this name, Ajinomoto. That is the salt, that is the powder which is always given in Chinese food. And also the KFC restaurant uses this kind of monosodium glutamate salt. So this monosodium glutamate salt which is used in the Chinese food or Chinese restaurant, you know, it has a very peculiar type of sensation, delicious kind of sensation. And because of this delicious nature of this taste sensation, the umami word is given. The umami is a Japanese word. The meaning of this umami is nothing but delicious, because of which the taste of monosodium glutamate is nothing but known as the umami taste sensation, right? It is detected by which type of receptor? Metabotropic receptor. And salt and sour, they are detected by which type of receptor? Inotropic receptor. Now, what is the name of this inotropic receptor? What is the name of this inotropic receptor? Or is there any difference between the G protein coupled receptor between three of them? Answer is yes. So again I have to remember a little more detail about these different ion channels, right? So look at inotropic receptor, that is the ion channel receptor. So this is nothing but a channel receptor. And metabotropic receptor means this is G protein coupled receptor, GPCR. Now look at salt and sour, this is the inotropic receptor. So for the salt, which ion channel is responsible? Epithelial sodium channel. Only one word you remember. For the salt is the taste of NaCl. It is detected by which ion channel? Sodium channel. What is the name of that sodium channel? Epithelial sodium channel, which is also present in other part of the epithelium surface. You know, we have discussed in detail in my app. Okay, so epithelial sodium channel is responsible for sense of salt. Sour, H plus ion. Genome has not updated this thing. H plus ion is mainly detected by a ion channel which is known as transient receptor potential channel P3 type. That is the main channel which is responsible for detection of this H plus ion. Getting my point? So sour taste is the taste of HCl, H plus ion, proton. Whenever you give proton, which ion channel is modified or activated? That is the TRP-P3, that is the transient receptor potential channel P3. Although H plus ion can also affect the INAC channel, HCN channel, you know that HCN channel, that is the hyperpolarizing induced CAMP nucleotide gated channel, which is also present in the nodal tissue of the heart, and it can also affect the acid sensing ion channel, which is also responsible for pain perception in response to chemical. So these ion channel are also present at the level of this dorsum of your tongue, right? They can be activated by sour, but the main channel which is responsible for this sour detection is nothing but your TRP-P3 ion channel. That all you have to remember about these two things. And believe me, if you remember this thing, you can answer any of the MCQ, whatever may be. This may be harder or easy, you can answer any of the MCQ. On the other hand, look at the metabotropic receptor. So metabotropic receptor means they are the G protein coupled receptor. Two family of G protein coupled receptor you have to remember. Two family. One is the T1 family and one is the T2 receptor family. Okay. So umami taste sensation, that is the sense of monosodium glutamate or Ajinomoto salt, they are detected by T1R1 and T1R3. And sweet sensation is also detected by T1R2 and T1R3. So I can say that this R3 is the co-receptor for both of them. The R3 is common. Okay, T1 R3 is common for both of them. That is the co-receptor for both umami and sweet sensation. But sweet is particularly detected by this T1R2 and umami is particularly detected by T1R1 family of G protein coupled receptor, which is a variety of metabotropic glutamate receptor. That's why I have used this word M. M for metabotropic. GLU means glutamate, and R4 type of metabotropic glutamate receptor, which is nothing but in the family of T1R1 and T1R3. And beta test, it is nothing but T2 family of G protein coupled receptor. So these three are the detail of this test receptor which are present on the test cell. Now after this nothing is remaining, only one thing is remaining, that the pathway for this test sensation from the surface of the tongue, right? So this is not very difficult because no one is going to ask you question from here, but we have already discussed a little bit that chorda tympani branch of facial nerve from anterior 2/3 and this glossopharyngeal nerve is from the posterior 1/3 of the tongue. All of them are reaching at the level of this nucleus tractus solitarius. Okay, from there another neuron is originating and it is reaching at the level of thalamus. Sometimes they can ask you that which nucleus of thalamus is responsible for this test pathway. You have to remember this is nothing but VPM, ventero-posteromedial nucleus of thalamus. Ventero-posteromedial nucleus of thalamus. Pain, temperature, touch, crude touch, pressure, everything that is mainly by the ventero-posterolateral, but here it is the ventero-posteromedial nucleus of thalamus. And from this ventero-posteromedial nucleus of thalamus, it is entering at the level of cortex. This is one thing I have to remember here, that the cortical area which is responsible for detection of your taste sensation is two important area. They are anterior insular cortex and the frontal operculum cortex. Anterior insula and frontal operculum. So these are the cortical area which are responsible for perception of your taste sensation, right? So this is all about today's class. This is the brief discussion of your smell and taste. Now you tell me whether you understood this topic or not. Okay, so this class is the starting. Okay, we are planning more such classes in the month of September through this Supermed channel. And what we are going to do in those classes, we are going to give you some MCQs in my Telegram channel. Also the MCQs will be posted in the Supermed Telegram channel. Followed by there will be discussion of those MCQs, and those MCQ will be particularly targeted for AIIMS entrance examination which are coming. After this is done, we will also target the NEET entrance examination, this MCQ detection, followed by their YouTube discussion through this Supermed YouTube channel, right? The MCQ discussion will be according to the topic which has been divided in my app. That means first we are going to discuss the general physiology, then we are going to discuss the nerve muscle physiology, followed by next topic, like that. Okay, so 15 to 30 MCQs will be given first, then you try those MCQs, then in the evening, maybe on the same day, maybe on the next day, we are going to discuss those MCQ in detail in this YouTube channel. Okay, so this is all about today's class. Thank you very much for listening, all of you. Thank you so much, Dr. Kiran. Thank you so much for your wording. Dr. Jyoti, thank you so much. MCQ, yes, that I am going to say that we are planning this thing. Okay, the Supermed has already given your answer, so thank you so much, all of you. Okay, so any queries or question from here? Okay, otherwise I am just switching off. Okay, right, so I hope there is no question. So thank you so much. If you have any queries also, you can directly connect me in my Telegram group, or you can connect me in the Facebook. This is all for today. Thank you so much.